If aging is the central mystery of medicine, then measuring aging must be the next frontier. For decades, we've measured age with the calendar, your chronological age, the number of years since you were born. But here's the uncomfortable truth. Two people can both be 60 years old, and one is running marathons while the other is managing things like diabetes, heart disease, and cognitive decline. So the same chronological age, but very different biology. So the obvious question becomes, is there a way to measure how old your body actually is? Not your birthday, not your appearance, and definitely not your step count, but your biology. That's where biological aging clocks come in. You've probably heard bold claims like reverse your biological age, drop your age by 5 years, or track your longevity. But what are these clocks actually measuring? And how are they built? What do they predict? And perhaps most importantly, are they ready for prime time and clinical decision making? Today, per usual, we're going to slow this way down. We're going to separate signal from marketing, mechanism from application, and certainly promise from proof. Because biological aging clocks may be one of the most powerful tools in geroscience, or perhaps one of the most misunderstood. Welcome to the Reprogram. And remember, knowledge is power. Hello, everyone, and welcome to the Reprogram podcast. I'm your host, Dr. George Murphy. Today, we have on the podcast Dr. Nicholas Skivir, who is an expert on the building and application of biological aging clocks. Nick, welcome.
Hi, good to be here. Thanks for having me.
So Nick, I think as a disclaimer, we should say that we know each other. Nick is actually a fellow in my laboratory here at the Center for Regenerative Medicine. So Nick, maybe you can give us a little bit of your origin story, how you became interested in biological aging clocks and measuring aging for that matter.
Sure. So one interesting thing about my relationship with George is that it actually extends back to before I started grad school. I was working in this lab after I did my master's degree, and this whole center here studies regenerative medicine and works with induced pluripotent stem cells. And just very briefly, it's 2026, and scientists now can take adult cells, usually blood, revert them back into stem cells, and then differentiate them into any cell type in the human body with the purpose of modeling disease and patient-specific medicine. And back when I worked here before grad school, I thought this was all very cool. Something that you actually mentioned to me kind of offhandedly back in the day, as you like to say, is that during this reprogramming process, the age of these cells are actually kind of functionally reset. The telomeres extend. Everything else that you can sort of quantifiably measure tends to be more kind of fetal in nature. So when I went off to grad school, this kind of stuck with me, and I thought it was very interesting. So when I was looking at labs to apply to work in, I saw aging labs, and I thought, this has a lot in common with regenerative medicine and has a lot of overlap with that. So that's kind of how I got into the field of aging.
Okay. So let's roll things back and let's start simply for our viewers. I think everyone has heard of biological aging clocks. These are things that are all over the internet. And I think one of the ways that we can introduce the topic is to compare or define the differences between biological and chronological age. So if you would, could you take us through the differences or how you would define biological versus chronological age?
Sure. Well, chronological age is very simple. This is the one that everyone is familiar with. We measure it by how many times the Earth has gone around the sun. We call them years, call things days. Biological age is a little trickier. It's more physiological in nature. It's kind of this umbrella term that covers a lot of different types of physiological decline and other things that happen with age, things like blood pressure, cognitive decline, things like that would all kind of fall under the umbrella of biological age. So it's a little bit broad and a little nebulous to define too. But it correlates very highly with chronological age, but it's fundamentally different. You would have two people that are the same chronological age, but they might have dramatically different physiological states. You might have one in a wheelchair and one that's running marathons who are both 60 or something. So that's kind of the difference in the way that we look at that.
So we could consider, chronological age, that's what it says on your birth certificate. Biological age, maybe that's what your molecular register, what your physiology is sort of telling you. So if someone had, a higher or lower biological age, what would that sort of look like practically?
So it depends on the model that you're looking at, but in general, if you know someone's chronological age and their biological age shows up as higher than expected, A lot of people would say that that correlates to an accelerated biological age. Conversely, if it's lower, then, then that's good. It kind of depends on the model, though. There are models that predict chronological age much better than biological age, and there are models that predict biological age better than chronological age. And there's this sort of trade-off that comes between them. So in theory, if you had a clock that was able to perfectly predict chronological age, in that case, you have no biological insight, right? If you're 45 and the clock every time you run it says you're 45, there's no room for variability there. It's just a perfect predictor, but you're losing all this biological information. So there's a sort of trade-off.
Yes. So I think what we've talked about on previous episodes of the podcast is that aging is like the Matrix. You can't really define what it is, and you can't be told what it is. You have to sort of experience it, which we do on a daily basis. So with that said, are you a believer in the fact that we can actually quantify or measure aging? Is that something that you think is possible in this current day and age?
Yeah, I think, aging is an incredibly complex and multifactorial thing that's happening. So we can certainly, as things stand, quantify different aspects of aging. Whether or not we can quantify the entire thing as a whole, um, it's still being defined, right? So that's kind of the dream in the future of the field. But I think right now we're getting certainly bits and pieces of it, but it's a very complicated thing to try to quantify.
No doubt. Okay, so again, we had talked about this concept, and the whole episode is on biological aging clocks, right? And so I think people are fundamentally familiar with these things or have heard about them on Instagram or on social media. One of the things that I would say is buyer beware. I think you might agree, a lot of other people in the field agree that the commercially available biological aging clocks or readouts that you can get via Instagram and other companies are not scientifically proven or grounded biological aging clocks, right? The kinds of things that we run in the laboratory are much more comprehensive based on data. So if you would, could you take us through how a biological aging clock is constructed, or sort of what are the underpinnings of sort of make it work? Can you run us through the different ways in which one of these might be constructed and applied?
Sure. There are a lot of different types of aging clocks. You can use generally whatever kind of omics data that you want, transcriptional, proteomic. We tend to like clocks that are based on methylation data. And just very quickly, methylation is you have different nucleotides in your genome, A, T, C, G. At the points where you have C's and G's that are next to each other, those can be methylated, which is just a methyl group being added onto it, CH3. And we have these fancy arrays that we can run to profile all these different millions of sites throughout the genome to see what the methylation states are. So whatever data that you're interested in, if you take thousands and thousands of samples with known chronological ages to go with them, you can train models using machine learning to take new methylation data and then spit out a chronological age or whatever you train the clock on, as an end product. So
And I think you're a proponent of epigenetic aging clocks. If you can give us a little bit of background, you really elegantly and simplified very well for the viewer what methylation data was and how we fuel these epigenetic aging clocks. But why is your preference, and why maybe of the field is the preference for epigenetic aging clocks historically and maybe even now?
yeah, yeah, there's a few different reasons. Methylation tends to be a lot more stable than things like transcriptomic data. So there's that kind of aspect. The data that we have for methylation tends to be very comprehensive. There's a lot of it. It's fairly high throughput. There are a lot of different types of arrays, but usually we use these ones from Illumina that profile about 1 million different sites. And in addition to that, they tend to be very well validated with other labs. A lot of people use them. They're applicable across all mammals at this point. People have made pan-mammalian clocks. So they tend to be very reproducible, and we like them for that reason.
And I've heard you in many of your talks and discussions, the origin of the epigenetic aging clock and our friend Steve Horvath. If you might tell us a little bit about that.
Oh, yeah. A little over, I think 10 years ago now, Steve Horvath was one of the people who first conceptualized this idea of using methylation arrays to create age predicting models. And he and a couple of other people around the same time came out with the first 3 models. His was kind of unique because it is a pan tissue predictor. So his worked across nearly all different tissues throughout the body because he trained it on a whole bunch of them. And it was kind of controversial at the time when it came out, but pretty quickly gained traction. And now there are tons of these different models, tissue-specific types for all different organisms. They're becoming very ubiquitous in the field. they've kind of exploded lately.
Yeah, and definitely, I think we both feel the same way. Shout out to Steve Horvath, who, in addition to being a brilliant scientist, is also a really fun guy and as good as advertised. So, hello, Steve, and thank you for all your work. So, these clocks, right? We have the ability to build them from pretty much any data. As you said, there's numerous layers of omics data, including methylation data that we can actually use. People have used, functional profiling. You can build a clock based on frailty data or your ability to sort of grip something very strongly. So there's lots of different ways in which you could plug in these data to sort of estimate someone's biological versus chronological age, right? And so I think, as you've elegantly described, you'd ideally want a biological age that's younger than your chronological age. And maybe that is indicative of you having more years of health span in front of you or less disease, morbidity, or mortality. These are the sort of pie in the sky things. But one of the things I think the field struggles with is what actually makes these clocks tick. So, in other words, if you have any type of clock, if somebody runs an epigenetic clock on their blood and they have a predicted biological age, is that something that's changeable, malleable? And what are the elements or what are the things that can actually make that clock tick or maybe even rewind itself? Can you give us some examples of things that might impact someone's biological age?
Yeah, so certainly, like I said earlier, there are a lot of different models. So it sort of depends on which models you're looking at. But in general, the earlier models tend to measure chronological age because that's what they were trained on, is just methylation data and chronological age. The later generation clocks, they tend to incorporate things like clinical data alongside that. So every clock that exists is sort of predicting a different proportion of chronological and biological, like a mixture of the two. As far as we can tell, like, biological age seems to be something that you can modulate with things like caloric restriction, healthy diet, exercise, things like that can modulate biological age.
What about other pharmacologic interventions, things that people take to try to rejuvenate themselves or to lower their putative biological age, or things like senescence or,
Oh yeah,
cell status?
yeah,
Yeah.
things like rapamycin and those type of things that kind of mimic caloric restriction can modulate that as well. There are things like mitochondrial changes, differences in stem cell populations, and nutrient sensing can have effects on the clock. But then things like senescence didn't, and that's not to say that like other clock models wouldn't be perturbed by those things. But I think every model is sort of capturing different facets of what it means to age. So like every clock is sort of measuring, I think, a mixture of different things. Certain clocks have been shown to measure things like to pick up on things like differences in cell type proportions, like differences like that, inflammation changes. But I think every clock fundamentally is kind of picking up on a bunch of different subsets of things. So it's not super clear cut to define. They're very accurate at predicting things, but interpreting them can be a little difficult.
And I think that's one of the reasons why, whenever we apply a clock model, we don't just apply one model. We basically look across an array of models to try to sort of come to consensus as far as what might or might not be happening. But I think the tantalizing thing here, and I think this is what our viewers are interested in, is the fact that maybe you have this new mechanism to test whether an intervention, a lifestyle change, you started to exercise more. Maybe you're worried about your metabolic health. Maybe a biological aging clock is a way that you can track these things, suggesting that what you're doing in your life, what you're taking, how you're treating yourself might be impactful and causal to these changes that might make the clock tick or maybe even decelerate, right? So I think one of the tantalizing things is to think about these things in the context of clinical medicine, right? So when we go to the doctor now, we run a series of really simplified blood tests, complete blood count, things like your hemoglobin, your hematocrit. But maybe we're approaching a time where a little bit of your blood might be plugged into a biological aging clock to see how you're doing at that predicted time. So, what do you think the possibilities of these types of instruments being used in the clinic, right? Is there any precedent for that actually taking place? Do you think it's a worthwhile thing? How far are we away from having a little bit of your blood being put into one of these molecular biological aging clocks to see sort of what your lifestyle is like?
Yeah, I think it could definitely be something that could be useful to look at in conjunction with other kind of canonical things. I think there's probably work that needs to be done in terms of just how to interpret this. Like, I think a lot of attention needs to be paid to What is the standard variation of these clocks across a population level? What kind of confidence intervals would we need to look at before we would see changes and say that, maybe this is indicative of something? I think there's just kind of a lot of testing that would need to be done, but certainly a lot of these models like pheno-age and grim-age and these types of things, they correlate very highly with different morbidities. And so I think that they could, in some cases, be useful diagnostic or predictive tools for incidents of that kind of thing.
So, as I alluded to in my open, one of the things that people talk about or things that are advertised to the general public are things that'll roll back your age, rejuvenate you in some concept. And we know, sort of as a field, that these things are not yet possible. But one of the things that you mentioned and is in featured in one of our earlier podcasts in our stem cell episode is the concept of creating an induced pluripotent stem cell, a master stem cell that can be made from your blood or skin cell that is a cell that then is imbued with the ability to create any cell or tissue type in the body. What we know, and this is something that we've experimented with in the lab, is that if we go through this reprogramming process to make this master stem cell from you, that at least at the cellular level, age reversal is possible. And this, as you noted, is something you can actually see in a biological aging clock. So you can actually see someone's blood, say that person is 55 years old, going through the reprogramming process to create a stem cell. And that stem cell is actually rejuvenated. It actually has not the age of the individual from whom it was taken, but now is the age of that person at the embryonic or fetal stage. So I think in your work now, one of the really cool aspects is using models such as this to try to understand really what makes these clocks tick.
Yeah, one of the things that we kind of noticed in our research was obviously that after age is reset during reprogramming, goes back to zero, but it can be kind of hard to reinduce or reinstill an epigenetic landscape onto these cells, which is kind of one of the reasons that using IPS models to study aging can be considered difficult by some people. And so in the course of this, we tried a whole bunch of different stressors and things to maybe kind of modulate that. And one thing we noticed is that specifically, like a couple of different types of organoids tended to show a significantly elevated epigenetic age relative to a lot of other IPS-derived materials that we've used. And kind of drilling down into this more specifically, we have labs in our center that study specific disease models. We profiled these as well with our epigenetic clocks and showed that a lot of these kind of mutations can result in a modulation of age as well, not just an increase of age, but in some cases a decrease of age if the mutation results in a kind of developmental delay and those kind of things, which we thought was very interesting to find.
Awesome. So basically, I think these are potential new tools that might have applications across basic science and emerging science, as we just discussed, but also tantalizingly, maybe in clinical practice, where you'd have a metric to understand how a particular intervention might work in someone or what someone's basic status of health actually might be. So if you had to summarize what clocks currently do well versus what they don't do well, how would you categorize those elements?
I think taken very broadly, clocks are very good at predicting what you train them to predict, right? So if that's chronological age or morbidity or lifespan, they're very good at that. But like because we don't have a really great definition of what aging is on the whole. If you ask 20 different people, you might get 20 slightly different answers. Um, right now clocks are kind of just proxy measurements for age because there's no gold standard definition of age that we have. So they're very good at predicting what you train them to predict. They're they can be very difficult to interpret biologically. So you were talking a little bit earlier about causal things. So, you might have some intervention where you see a change in an epigenetic age and you don't know necessarily whether this is something that is a driver of age or whether this is just a result, right? If you roll
Sure.
back the odometer in your car, it doesn't mean that you have a newer car. So, it's possible that this is the case with some clock models as well. It's also possible that they can give insight into the biology of aging and the mechanisms of aging if they are causal. But this is something that I think people are currently investigating in the field. There are some newer clocks that purport to be kind of causal clocks and that type of thing by using specific sites that are shown to correlate with morbidity and things like that. So that's certainly very interesting to look at, and I think people will continue to make a lot of discoveries in that avenue. in the future.
But perhaps right now, particularly in social media, maybe the marketing is outrunning the science. But maybe in a perfect world, somewhere down the line, and I think there's a lot of potential for these types of tools. If we could perfectly measure biological age, if we had the perfect set of systems or a combination of clocks that could do this, how do you think that would change medicine? Would that change treatment regimes, the way in which you go to interact with your general practitioner? How would perfect prediction via biological aging clocks change medicine, do you think?
It would be amazing because firstly, if we can perfectly predict biological age, that means we've also perfectly defined biological age, right? So if we can do that, I think for things like clinical trials, first of all, it gives you a really good surrogate endpoint. So you can simply just measure with the clocks whether the age has gone up or down, and you can say, boom, this intervention is effective at slowing aging or not. And then secondly, I think, this would become something ubiquitous when you go to a doctor's office. You would measure your biological age every time. You could tailor it towards, more patient specific medicine. So, if you have two different 60-year-olds, you could make informed decisions about maybe this one can undergo invasive surgery and have a high probability of being resilient enough to recover from it versus this other one might not based on their biological age and things like that. So it would be hugely impactful, I think.
Fantastic. And maybe lastly, have you made any changes? So, as far as the aging research that you've done, and we've tested your biological age, we've tested my biological age, and we keep sort of running metrics of these in the laboratory. Have you made any changes or thought about interventions in your own life based on some of the data that you generated from yourself?
I sort of practice caloric restriction, but I've sort of always done that just by default. I just don't get like hungry that often. So, I eat a couple meals a day, usually maybe like 1.5 meals a day. But so hopefully that's good. I mean, I've measured my biological age. It's pretty much like where it's supposed to be. But I don't have a whole lot of time points, so I'm not sure like what their trajectory is. Maybe it's good. I hope it's good. But
Yeah, sure. So I think right now marketing maybe is ahead of the actual practical application of these clocks. But as we've discussed, fundamentally they have a tremendous potential upside, both in basic science, but also maybe in the clinic as far as being able to interpret potential interventions, or as you noted in clinical trials. You can't really run a clinical trial on something that you expect to be a geroprotector and wait 80 years to see if the person lives longer. Maybe this could be a proxy of something having an impact. So certainly, real world potential that could be impactful all over the place, but maybe buyer beware at this point. So I guess lastly, would you recommend people going out and having their biological age tested to try to understand maybe their molecular underpinnings or maybe potential drivers of age or disease in their systems?
I think it's something right now that's currently it's cool to look at. It's cool to see. It's hard to make. I wouldn't make any like broad sweeping decisions based off of it. But I think it's getting there. I clocks are so popular now that there are dozens of new models that come out every single year, and I think that as we move forward, they will get there. It's interesting because if you look at clocks, the clocks that are the very best at predicting chronological age, there's one called the Zhang clock. They tend to be looking at a lot more degree of like stochastic variability than the biological age
Can you
predictors.
define that for your general citizen, stochastic
Just kind of like
variability?
biological noise in your genome. So when you're young, your genome is very organized and put together. And as you get older, the regions of your genome that are closed and open kind of tend to kind of regress towards the mean a little bit. So it becomes less organized, and there's more noise in all of the signal that you have. So you're in terms of like gene expression, methylation, whatever you're looking at, it will be noisier. And I
So
think
we love that concept in lab, right? The concept of entropy, right? I love this concept of entropy. And the way that we can potentially think about it is as you age, maybe as you go through damage accumulation, there's more noise in the system, an increase in entropy. And maybe that's something you could measure, and that could correlate to the degree of damage or sort of how you're progressing through life, the degree of disorder and disarray in a system. And you are a system as a human being, and that's something that you could potentially read out.
right, right. And like I said, that tends to be something that the more chronological clocks pick up more than the biological ones. So it's kind of an interesting kind of a trade-off there because they're certainly all different facets to the aging process.
Yes. And I think the tantalizing thing, as far as the potential of these clocks, and I think what the general consumer is interested in, is that higher or lower biological age is not just something that can tell you something interesting about maybe your lifestyle, your exercise regime, things that you're actually taking as potential rejuvenative interventions, but maybe it's something that's malleable or changeable, if you will, right? If you make a certain change in your life, you want to know if that's going to be impactful. And maybe in the perfect world, this readout would be a biological aging clock.
Yeah, they're very convenient because they can kind of distill all this very complex biology into just a nice manageable number that you can kind of make relative comparisons to. Um,
Any thoughts? So, obviously, there's a lot of longevity influencers who talk about their decelerated aging rate, right? Any belief in that? So, if you're a longevity influencer and you have the lowest biological age or the slowest rate of aging, do you think that that's something that's really meaningful and something that can be quantified as a metric to how well you're aging?
yeah, I think it's really it's interesting. Certainly, like the studies that purport to have come up with like aging the clocks that measure the rate of aging, like Dunedin and Pace. There are these studies that kind of use this longitudinal data where they're looking at the same people over the course of decades and kind of looking at their retroactively, like looking at their health outcomes to kind of try to predict the rate that they're aging. So it's kind of like a speedometer at any given point instead of just like a static measure of how old you are. And I think this is a very cool concept. But again, I think it's very difficult to interpret like what it means if you have a lower one. Is it the number is lower, but is it what does it mean biologically? It can be very difficult to interpret. Everyone is sort of different. I think everyone ages a little bit differently, even from tissue to tissue within your body. Those can age at different rates. So if you're a smoker or something, your lungs are probably aging faster. So I think longitudinally though, it's very powerful. It's not data that's like super easy to come by, but I think a lot of people are looking at that now, and I think in the future this will only empower the generation of better and better clock models.
Yeah, and so as you noted, there's many, many different clock models, including those that are organ specific. So maybe you have older lungs and a younger kidney. I always say to myself, well, maybe if we come up with the ability to rejuvenate one tissue type, then you're still going to sort of die at the same age because you're going to die of something else. You open up the ability to then develop disease in a different organ or tissue type. So maybe it's a zero sum game. It's hard to tell, right?
Yeah,
Yeah.
definitely, absolutely. It's certainly going to become something that will become a much more informative tool, I think, in the future.
Awesome. Nick, it was fantastic to have you on the podcast. I
Like,
appreciate all of your efforts and a lot of the discoveries that you made in lab, and I can't wait to see the next generation of those discoveries. Thanks for being here.
thank you. Thanks for having me.
So, in closing, biological age may tell us how well we're living, but knowing that number is only powerful if we understand exactly what it means. This is the Reprogram, and this episode is about measuring time biologically. I hope you enjoyed this discussion, and please follow our podcast and join me on this journey of understanding and self discovery as we navigate our way into the fields of longevity, aging biology, regenerative medicine, and the emerging field of geroscience.